You think you know weird. You think you've seen strange. Down in the abyss, where sunlight dies at 600 meters and pressure crushes steel like paper, things get biblical. We're talking about a zone that covers more than half of Earth's surface, 3.5 million square kilometers of the Pacific alone, and most of it has never been touched by human eyes. October 23 to November 28, 2024, Garanstes. The research vessel Meteor ran expedition rubble in the Vima fracture zone. They found creatures that shouldn't exist, transparent heads filled with rotating eyeballs, fish that fuse their bodies together for sex, crustaceans that can go 5 years without eating, animals that glow in pitch darkness 4,000 meters down, every single one built for a world we can barely reach. If you value documented deep sea biology and extreme adaptation history like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual, using sources from the Senckenberg Research Institute's Ocean Species Discoveries platform, published November 2, 2025 to hours, and the Monterey Bay Aquarium Research Institute's Barrel-Eye Fish Studies from 2008, led by Bruce Robison and Kim Reisenbichler. Now back to the information. The abyss starts where the twilight zone ends, 3,000 meters. That's where the abyssal zone begins and runs to 6,000 meters deep. No light, 1.5 degrees Celsius, 800 times atmospheric pressure, and it's not empty. March 20, 2024, the UK research ship James Cook wrapped a 45-day expedition to the Clarion-Clipperton zone between Mexico and Hawaii. They photographed over 30 potential new species: glass sponges, bristingid sea stars perched on sponge stalks, sea cucumbers nicknamed the unicumber because you can see through their bodies, pink sea pigs the size of softballs. The Natural History Museum team found more biodiversity down there than anyone predicted. 6,000 to 8,000 species waiting to be described. According to Current Biology, June 2023, start with the builders, the glass sponges, Hexactinellida. They grow on polymetallic nodules that take millions of years to form, potato-sized mineral chunks scattered across the Clarion-Clipperton zone, covering an area the size of Australia. The sponges are made of silica, the same stuff as glass, arranged in geometric lattices that can live for thousands of years. They filter marine snow, dead algae, fecal pellets, mucus from the endless dark column above. One sponge can process hundreds of liters per day. The abyssal plain has more biomass in the top 1 centimeter of mud than in the entire 4,000 meters of water above it, according to Natural History Museum surveys from March 2024. Why? Because everything denser than water sinks. For 180 million years, marine snow has been falling on parts of the Pacific. That's Diplodocus time, ichthyosaur time. The deep sediment layers prove it. Then the movers, sea cucumbers, Holothuriidea. They're the vacuum cleaners, the earthworms of the ocean floor. At depths past 8,900 meters, sea cucumbers make up 90% of total macrofauna mass. They eat their way across the seabed, ingesting sediment, extracting organic matter, excreting the rest, bioturbation, reworking sediment, oxygenating it, recycling nutrients. The species vary wildly. And Parama, the Barbie pig, pink, tubular, walks on stout legs with elongated mouthparts to select detritus. Cycropodes longicauda, the gummy squirrel, 80 centimeters long, bright yellow body, red underbelly, 18 feeding palps around the mouth. The standout feature, a massive dorsal sail held upright like a squirrel tail. Scientists from the Deep Sea CZ expedition in June 2018 think it catches weak deep-sea currents, letting the cucumber bounce along hunting better feeding grounds. The yellow might signal toxicity to rat-tail fish. Nothing eats them, even though they look like lime gummy bears. The transparent ones are stranger. The unicumber, family Elpididae, genus Penygone or Amperima, clear skin. You can see the intestine packed with yellow sediment through the body wall. Large tube feet, a small velum, a sail on the dorsum. They hover and plow through mud at depths past 5,000 meters, and they move in herds. Congregations of tens to hundreds crossing bathygraphic features of the abyssal plain like migrating herds on land. Now the scavengers, giant isopods, Bathynomus giganteus, crustaceans, relatives of pillbugs and roly-polies. Except they reach 50 centimeters long and weigh nearly 4 pounds. Deep sea gigantism, when animals grow far larger in the abyss than shallow water cousins. The giant squid does it, hitting 60 feet. Regular squid stop at 2 feet. Why? Theories stack up. Kleiber's law says larger animals have more efficient metabolism, basal metabolic rate scales to roughly 3/4 power of mass. Under food scarcity, that matters. A giant isopod, kept in Japan, reportedly survived 5 years without eating. 5 years? Their metabolism is that slow, eh? One study from February 2017 in Animal Behavior measured Bathynomus giganteus at a routine metabolic rate of 21.8 milligrams of oxygen per kilogram per hour at 13.4 degrees Celsius. That's glacial. They live 365 to 2,140 meters down, mostly. Bathynomus kensley goes to 2,500 meters. They prefer mud or clay seafloor, solitary lives, but when food appears, a whale carcass, a dead fish, squid remains, they gorge. They'll distend their bodies to the point they can barely move. A meal at 45% of body weight increases metabolic rate 6.5-fold for 20 days. Then they wait. One isopod was filmed attacking a dogfish shark in a deep water trap, latching onto its face, eating it. Temperature matters. Bathynomus doderlini stops feeding below 3 degrees Celsius. That temperature limit explains why they're absent from polar regions where abyssal waters drop colder. The shell is armor, overlapping calcareous segments. The first segment fuses to the head, extends over the eyes. The eyes are compound, over 4,000 facets, giving wide fields of view and sensitivity to fast movement. They have a tapetum lucidum, the same reflective layer cats and alligators use for night vision. It sits behind the retina, reflects light back through, creates a glow, but vision is still poor in near total darkness. 14 legs, little hooked claws at the tips for stability on the seafloor. No ink sac, no chromatophores for color change, just a thick chitinous shell and patience. Reproduction is slow. Females develop a brood pouch. Marsupium, when sexually active, holds up to 30 eggs, each half an inch, the largest eggs of any marine invertebrate. Males have 2 organs, penis, small white appendages carrying sperm, and an appendix masculina, which transfers sperm. Smaller isopods usually have bigger penis. According to Aquarium of the Pacific Keepers, after the female's spawning molt, the male injects sperm while she's softer. Then she buries herself in sediment until the eggs hatch. Slow metabolism becomes a feature, not a bug. Alphonse Milne Edwards, French zoologist, first described the genus in 1879 after Alexander Agassiz collected a juvenile male Bathynomus giganteus from the Gulf of Mexico. Before that, scientists believed the deep ocean was azoic, lifeless. Sir Charles Wyville. Thompson's work on the Challenger expedition proved otherwise. From October 23 to November 28, 2024, the Meteor expedition sampled over 6,000 meters in the Atlantic's Vima fracture zone. Isopods were there, diverse. The Clarion-Clipperton Zone surveys show they're one of the most dominant macrobenthic groups. Different species at different depths. Deeper water means fewer species, but larger individuals. Now the hunters, anglerfish, Ceratioidae, 170 known species and 12 families. They live 1,000 to 4,000 meters down in the bathypelagic zone. Pitch dark, matte black skin that absorbs 99.96% of light. According to Current Biology, July 2020, studies on ultra-black camouflage. The skin is packed with melanosomes arranged in specific structures that trap photons, makes them nearly invisible even with bioluminescent searchlights. The females are the monsters, bucket-sized mouths, razor teeth angled inward. When something's in, it's not getting out. Tiny frosted eyes. Spiky fins, bodies shaped like blobs, designed for drifting, not chasing. Size varies wildly. Melanocystus johnsoni is squat and round. Thaumaticthys binghami is flat with a huge snout. Colophrine jordani is covered in whiskery filaments. They all share one thing, the esca, the lure. A modified dorsal fin spine that dangles in front of the mouth like a fishing rod. Inside the esca, bioluminescent bacteria, Photobacterium or Vibrio species. The bacteria make their own light through chemical reactions, luciferin and luciferase. The anglerfish has no genes to produce the light itself. It acquires the bacteria from the environment. A study in eLife 2019 by Laura Baker and colleagues showed that diverse deep-sea anglerfishes share a genetically reduced luminous symbiont. The bacteria's genome shrinks over generations as they specialize for life inside the escae. The anglerfish can hide or reveal the lure using a muscular skin flap. By pulsing the light and moving the lure, they attract pelagic crustaceans, fishes, other prey. The female hangs nearly motionless in the water column. When something approaches the light, the massive jaws snap shut. Done. The anglerfish has an expandable stomach, so it can swallow prey larger than itself. But here's where it gets truly weird. The male serratioid anglerfish are dwarfs, a few centimeters long. They have enormous nostrils, weak jaws, degenerate guts. They're built for one purpose, find a female. They follow pheromones, scent trails across kilometers of empty water. When a male finds a female, he bites into her skin. His tissues fuse with hers. Blood vessels merge. His body begins to atrophy. Eyes shrink. Internal organs dissolve. Eventually, nothing remains but testes, fused permanently to the female's body, fed by her bloodstream. Parasitic males. In some species, multiple males can attach to one female. How does her immune system tolerate this? Research in Science, July 2020, by Thomas Bohm and colleagues, found that serratioid anglerfish lack the genes to produce most molecules that attack foreign tissue. They have few or no T cells and antibodies, no adaptive immune system. That would kill a human, but it's what anglerfish need for sexual parasitism. The female gains a permanent sperm supply. The male gets food and the chance to reproduce. It's a bizarre strategy for a bizarre environment. Current Biology, June 2024, published a phylogenetic study by Chase Brownstein and Thomas Neer, showing sexual parasitism evolved when anglerfishes invaded the bathypelagic realm. The transition happened fast. Multiple major lineages formed in quick succession. Speciation rate in serratioids is higher than in benthic anglerfishes. Parasitic males and the bathypelagic environment together drove an adaptive radiation. Anglerfishes became the largest evolutionary radiation of vertebrates in the midnight zone. The first anglerfish known to science washed ashore in Greenland in 1833. Johannes Christopher Hagemann Reinhardt in Copenhagen examined it. Hymentolophus groenlandicus, the football fish, the man gobbler. Since then, new species keep appearing. Gigantactus paresca, discovered by University of Hawaii oceanographers in the Clarion-Clipperton Zone during a 2021 cruise, made the World Register of Marine Species top 10 in 2024. Samantha Rickle published the description. It's another testament to how much remains unknown. Then the specialists, the barrel-eye fish, Macropinna microstoma, Opistheproctidae family, found off California's coast and throughout the North Pacific, Atlantic, and Indian Oceans, 600 to 800 meters depth in the mesopelagic zone, 15 centimeters long. Its skull is transparent. The head is a fluid-filled dome. Inside, two tubular eyes point upward, capped with bright green lenses. The green pigment filters sunlight from the surface, helping the fish detect bioluminescent glows of jellies and siphonophores directly overhead. Those two black spots at the front of the face that look like eyes. Nares, olfactory organs. Nostrils. The eyes are packed with rods. No cones. Maximum sensitivity to faint light. Barrel eyes hover motionless in the water using large, flat fins for precise maneuvering. They're looking up through their own heads, watching for silhouettes or bioluminescent prey. When they spot something, a drifting jelly, copepods, small crustaceans trapped in siphonophore tentacles, the eyes rotate forward. The fish tilts its body and swims upward in feeding mode. Bruce Robison and Kim Reisenbichler at Monterey Bay Aquarium Research Institute published this discovery in Copea, 2008. Before that, scientists thought the eyes were fixed. Specimens brought to the surface had their delicate transparent domes crushed by pressure changes and net handling. Robison and Reisenbichler used remotely operated vehicles to observe living barrel eyes in their natural habitat. They saw the eyes rotate within the transparent shield. The dome protects the sensitive eyes from the stinging nematocysts of siphonophores. Siphonophores in genus Apolemia grow over 10 meters long. Their colonial jellies trailing thousands of stinging tentacles like living drift nets, capturing copepods and small animals. Barrelays maneuver among the tentacles, picking off captured prey. The transparent shield takes the stings. The rotating eyes keep locked on the prize. Macropinna microstoma was first described in 1939 by marine biologist W. M. Chapman. It wasn't photographed alive until 2004 when Mbari researchers captured images in the North Pacific. A Russian study from 2016 by Nazarkin found fossil evidence showing genus Macropinna existed millions of years ago, traced back to the Miocene epoch. The tubular eyes are ancient adaptations, not recent. Barrel eyes reproduce as pelagic spawners. Females release buoyant eggs coated with oil that float to the surface. Males release sperm in the water. After hatching, larvae drift in currents at shallower depths. As they mature, they descend, developing the transparent dome and tubular eyes. Other barrel eye species in Opistheproctidae vary. Opistheproctus and Macropinna are stout and deep-bodied. Dolichopteryx and Bathylichnops are extremely slender, elongated. Rhynchohyalus and Winteria are intermediate fusiform shapes. All have large telescoping eyes enclosed in transparent soft tissue domes. All gaze upward but can redirect forward. The retina has exceptionally high rod cell density and rhodopsin concentration. No cone cells, pure night vision. Some species have bioluminescent organs on their undersides for counter-illumination, matching the downwelling light to erase their shadows when viewed from below. It's camouflage in a world where any silhouette gives you away. Now the survivors. The vampire squid, Vampiroteuthis infernalis. Vampire squid from hell, but it doesn't suck blood. It's not even a squid. It's the only surviving member of order Vampiromorphyta, a phylogenetic relict. Closest relatives are octopuses. It has 8 arms, but also 2 retractile filaments between the first 2 pairs of arms on the dorsal side. These filaments can extend to twice the animal's length. They're lined with tiny hair-like structures, sensory cells, that detect vibrations and particles in the water. The vampire squid uses them to capture marine snow. Detritus, fecal pellets, dead zooplankton remains. It's a detritivore, not a predator. It lives 600 to 900 meters down, sometimes deeper to 2,500 meters. In oxygen minimum zones throughout temperate and tropical oceans. OMZs are regions where oxygen drops below 2% saturation. Most animals can't survive there. The vampire squid thrives. How? It has the lowest mass-specific metabolic rate of all deep-sea cephalopods. Blue blood with hemocyanin that binds and transports oxygen more efficiently than other cephalopods. Gills with exceptionally large surface area. Weak musculature but sophisticated statocysts, balancing organs like human inner ear, and ammonium-rich gelatinous tissues that match the density of surrounding seawater, providing neutral buoyancy with minimal effort. The body is small, 30 centimeters max. The eyes are huge, proportionally the largest of any animal relative to body size. They're sensitive to the faintest bioluminescence. The vampire squid lacks ink sacs, no escape jet of black cloud. Instead, it has photophores, light-producing organs covering its body. When disturbed, it can eject sticky bioluminescent mucus from its arm tips. Thousands of tiny blue orbs hang in the water for nearly 10 minutes, dazzling predators. The light can stick to the predator, creating a burglar alarm effect, making the predator visible to its own predators. It's metabolically expensive to regenerate the mucus, so the display is a last resort. The vampire squid also has a defensive posture. If threatened, it curls its arms up and outward, wrapping them around its body, turning inside out. The webbing between the arms is heavily pigmented dark, concealing the photophores. Spiny projections on the tentacles, cirri, point outward. The glowing arm tips cluster together far above the head, diverting attacks away from vital organs. The arms regenerate if bitten off. It's a distraction. While the predator chews on an arm tip, the squid jets away with erratic movements and writhing glows, making it hard to track. With the long filaments deployed, vampire squids drift in the currents. If the filament touches something or detects vibrations, the animal investigates with rapid acrobatic movements. It can swim at 2 body lengths per second with 5-second acceleration. But weak muscles mean limited stamina, no prolonged flight. Everything is energy conservation. The vampire squid feeds by secreting mucus from the suckers on its arms, combining waste particles into balls of food, then transferring them to the mouth with the filaments. Confirmed stomach contents include gelatinous zooplankton remains, salps, larvaceans, medusae, and complete crustaceans like copepods, ostracods, amphipods, isopods, plus fecal pellets. Some mature specimens have fish bones and scales. Opportunistic scavenging. Reproduction is slow. Ovulation is irregular. Minimal energy goes to gonad development. Females produce a small number of large eggs, each 8 millimeters. Case-selected strategy. The eggs are thought to be buoyant and planktonic. No seasonal breeding pattern. The distribution of eggs is similar year-round. Males lack a hectocotylus, the specialized arm other cephalopods use to transfer sperm packets. Instead, they pass sperm directly with their large suckers. Carl Chun first described Vampiroteuthis in 1903 from specimens collected on the Valdivia expedition. He thought it was an octopus. Later studies placed it in its own order along with extinct taxa. The name Vampiroteuthis comes from Latin vampirus and Greek teuthis, squid. The genus has existed for millions of years, a living fossil persisting in an environment few predators can access. Giant grenadiers, deep-diving whales, sea lions, those are the confirmed predators from stomach content studies. Then, the deepest, Dumbo octopuses. Genus, Grimpoteuthis. 17 known species, part of suborder Sirena, the cirrate octopuses. They live 1,000 to 7,000 meters down in the bathyal and abyssal zones. The deepest confirmed sighting was in the Java Trench, 2020 dirt, at 6,957 meters. That's Hoddle Zone, the deepest part of the ocean. These are the deepest living octopuses known. They're named for their ear-like fins that protrude from the mantle just above the eyes. They flap these fins to swim, looking exactly like Dumbo the elephant. The body is gelatinous, bell-shaped, semi-transparent. Most individuals are 20 to 30 centimeters long. Although one specimen reached 1.8 meters and weighed 5.9 kilograms, the arms are connected by webbing. Umbrella octopuses. When they spread their arms, they form an umbrella shape. Each arm has a single row of suckers, about 65 to 68 per arm hours, plus cirri, finger-like projections along the arms. The cirri play a key role in chemoreception, handling prey and creating water currents to move food to the mouth. Grimpoteuthis have an internal cartilaginous shell, U or V-shaped, giving them structural support. No rigid skeleton like other octopuses. They lack ink sacs, no color-changing chromatophores. The skin is pigmented orange, red, or purple. In most Grimpoteuthis, the oral surface of the arms is deep purple, the rest pale or unpigmented. They have large eyes, but their vision is likely poor in total darkness. They may rely more on chemoreception and mechanoreception. They hover just above the seafloor using fins swimming, flapping the ears. They also use umbrella-style drifting, spreading the arms and webbing and floating neutrally buoyant in the currents. Takeoff mode is a sudden contraction of the brachial crown and web. Pumping mode is slow, using peristaltic waves through the webs. They are benthic creatures, living on or near the abyssal plain at depths around 3,000 to 4,000 meters. The average ocean depth is 3,790 meters. They're right there. Feeding hasn't been directly observed in Grimpoteuthis, but is presumed similar to Opistoteuthis, another umbrella octopus. They likely trap small prey items in the webbing, enclose the prey between the webbing and the seafloor or within the arm webbing, then use the cirri to move food to the mouth. Known prey from dissected animals includes benthic polychaetes, benthopelagic copepods, amphipods, isopods. They swallow prey whole. The radula, the ribbon-like band of teeth in cephalopod mouths, is reduced or absent in Opistoteuthidea family. Reproduction is continuous. Females carry multiple eggs in various stages of maturation. They lay 1 or 2 eggs at a time with no seasonal spawning. Mating has never been observed in serrate octopuses. Males lack a hectocotylus. Instead, the male uses large suckers to pass sperm packets, each within an operculum, directly into the female's mantle cavity. The female stores the sperm and uses it to fertilize eggs when ready. The eggs are large and have a tough casing surrounding the chorion, not found in other octopuses. Eggs emerge within a covering that solidifies on contact with cold seawater. Grimpoteuthis attaches eggs to deep-sea corals, octocorals. The female doesn't guard or incubate them. Hatchlings emerge fully competent with all sensory and motor features to survive alone. Dumbo octopuses have copper-rich blue blood, hemocyanin, more efficient at transporting oxygen in low temperature. Oxygen depleted deep sea water than hemoglobin. Enzymes adapted over evolutionary time to function under extreme pressure and cold. Chemical processes like digestion work at temperatures and pressures that would shut down shallow water animals. They face few predators in the deep. Some deep-diving fish, tuna, sharks, and marine mammals like dolphins hunt them in the upper part of their depth range. In the true abyss, predators are scarce. Grimpoteuthis was named by Guy Robson, former zoology department curator at the Natural History Museum in London. 2 specimens from the Challenger expedition are housed in the mollusca collection there. We don't have enough data to know if Dumbo octopuses are endangered, but deep-sea mining threatens them. Polymetallic nodule extraction in the Clarion-Clipperton zone will disturb the abyssal plain where they live and feed. Every ecosystem is delicate, especially habitats this specialized. Alongside Dumbo octopuses and vampire squids, the abyss holds other cephalopods, glass octopuses. Vitrillidonella richardi, nearly transparent. Only the digestive gland and eyes are visible. Glass squids, Crunchidae family. Transparent bodies, some with elongated coiled guts or organs positioned to minimize silhouette. They use counter-illumination photophores on their undersides to match downwelling light. Whiplash squids in family Mastigoteuthidae, long, thin, tentacular clubs for snaring prey in midwater. Then there are the jellies. Siphonophores dominate midwater. Apolemia species grow over 30 meters. Colonies of specialized zooids, each a clone, stringing together to form one functional organism. The head pulls the colony through water. Thousands of stinging tentacles trail behind, capturing copepods and small animals. Stygiomedusa gigantea. Giant jellyfish, bell diameter up to 1 meter, 4 arms up to 10 meters long. It moves through the abyss like a ghost, pulsing slowly, conserving energy. Catenophores, comb jellies. They're not true jellies, no stinging cells. They use 8 rows of cilia, combs, to propel themselves. Bioluminescent Bathycyro species found at 3,000 meters. Bloody belly comb jelly, Lampoctius cruentiventer, in the twilight zone. The red gut conceals bioluminescent prey so the comb jelly doesn't glow and attract predators. The crustaceans diversify into strange forms. Amphipods. Hirondelia gigas in the Mariana Trench, 10,900 meters. The deepest living crustacean, piezophilic, pressure-loving. Copepods scatter throughout the water column. Ostracods glow blue when disturbed. Giant ostracods like Gigantocyprus reach 3 centimeters. Massive for ostracods. They're predators, ambushing prey in midwater. The fish beyond anglerfish and barrel eyes include rat tails, grenadiers, family Macrouridae, elongated tails, bulbous heads, large eyes, benthopelagic. They cruise just above the seafloor hunting for isopods. Amphipods, small fish. Coryphanoids species reach 2 meters. Snipe eels, family Nemicthidae, extremely elongated bodies, needle-like jaws. They feed on small crustaceans. Gulper eels, Uropharynx pelechinoids, massive hinged mouths that can swallow prey larger than themselves. They live 2,000 to 3,000 meters depth. Chimeras, holocephaly, ghost sharks, ratfish. They have venom spines on the dorsal fin. Males have retractable sex organs on the forehead, tentaculum used to grasp females during mating. Spotted chimeras glide over the abyssal plain on pectoral fins like wings. Bioluminescent mucus coating the skin may deter parasites or predators. Then the oddities, the tripod fish, Bathypteroi species. They perch on the seafloor on elongated pectoral and pelvic fin rays, stilts. They face into the current with upward angled mouths, catching drifting crustaceans. Some are simultaneous hermaphrodites, both male and female gonads functional at once, an adaptation to low population density. When you rarely encounter another individual, being able to mate with anyone you find is advantageous. Xenophyophores, giant single-celled organisms, foraminifera. They build tests, shells, from sediment particles glued together with organic cement. Some reach 20 centimeters diameter. They're found on abyssal plains in the Clarion-Clipperton zone. Gooday and colleagues describe new species from the eastern CCZ and Zootaxa, 2024. These protozoans are the largest single cells on Earth. The sponges keep building. Cladorhizidae, carnivorous sponges. They don't filter water. They trap small crustaceans on hooked spicules, glass needles, then slowly digest them. Found at depths over 3,000 meters, some species have stalks that raise the body off the sediment into the current. Bryozoans, moss animals, colonial. They form branching structures on polymetallic nodules and hard substrates. Each individual, zooid, is millimeters long, but the colony can be decimeters. They filter feed using ciliated tentacles called lopophores. Sea stars diversify. Brisingida, many-armed, up to 20 arms. They perch on stalks, sponge bodies, raising their arms into the water to catch drifting particles. Arm tips can regenerate if damaged. Porcelanoasteridae, cookie-cutter stars, flat bodies, paddle-like arms for pushing through soft sediment. They feed on buried fauna. Corals persist in the abyss without sunlight. Octocorals, soft corals, they don't build massive reefs, but they provide habitat. Chrysogorgia species, golden coral, bamboo coral, Isididae family, alternating nodes and internodes, flexible organic joints, and rigid calcareous segments. They can bend in currents without breaking. They're suspension feeders, extending polyps to catch detritus. Some colonies are hundreds of years old. The abyssal plains themselves are ancient. Marine snow has been falling for 180 million years in some Pacific basins. The sediment is ooze, foraminiferal ooze and carbonate compensation depth above 4,000 meters. Radiolarian ooze where silica dominates. Red clay in the deepest zones where nothing calcareous survives. The accumulation rate is millimeters per thousand years. Slow, patient, burying everything, and the food web teeters on the edge of starvation. Primary productivity happens at the surface. Phytoplankton, sunlight, photosynthesis. Only 1% of that energy reaches the abyssal plain. It sinks as marine snow, dead algae, zooplankton molts, fecal pellets, mucus strands. The fall takes weeks. By the time it arrives, it's been picked over by mid-water animals. What remains is the lowest quality food, high in fiber, low in protein. But the abyssal community survives on it. Seasonal pulses matter. Phytoplankton blooms in surface waters, spring blooms, upwelling events, send massive amounts of fresh organic matter down. Monterey Bay. Aquarium Research Institute studies from November 2013 showed blooms of algae or animals near the sea surface can deliver as much food to deep-sea organisms as would normally arrive over years or decades. feast and famine. When a bloom hits, the seafloor erupts. Sea cucumbers converge, isopods swarm, fish gather, everything gorges. Then the desert returns, months or years with minimal input. Survival depends on extreme metabolic efficiency. Whale falls punctuate the starvation. When a whale dies and sinks, it becomes an oasis. A 40-ton gray whale carcass delivers 2,000 kilograms of lipids to the abyssal floor. The carcass supports a succession of communities. Mobile scavengers, hagfish, sleeper sharks, rat tails, arrive within hours, strip the soft tissue in days to months. Opportunistic enrichers, polychaete worms, crustaceans, mollusks, colonize the bones, consuming residual organic matter over months to years. Sulfophilic bacteria colonize the lipid-rich skeleton, chemosynthesizing energy from bone oils. The whale fall can sustain specialized communities for decades. Osedax worms, zombie worms, females drill into bone with acidic enzymes, extract lipids with symbiotic bacteria and root-like tissues. Males are microscopic, living inside the female's tube. Hydrothermal vents punch holes in the desert, mid-ocean ridges, seafloor spreading centers. Magma heats seawater circulating through cracks in the oceanic crust. Superheated water, 350 degrees Celsius, shoots out in black smokers or white smokers, precipitating minerals. Chemosynthetic bacteria oxidize hydrogen sulfide, methane, hydrogen for energy. They're primary producers without sunlight. Tube worms, Riftia pachyptila, at East Pacific rise vents, grow 2 meters long. No mouth, no gut. A trophosome filled with sulfur-oxidizing bacteria provides all nutrition. Giant clams, Calyptogena magnifica, yeti crabs, Kiwa hirsuta, farming bacteria on their hairy arms. Vent communities are dense, productive, completely disconnected from surface photosynthesis. But vents are ephemeral. Tectonic shifts close them. Eruptions bury them. Communities must disperse, colonize new vents. The abyssal plains are the highways between vent islands. Cold seeps are slower but longer-lived. Methane, hydrogen sulfide, hydrocarbons seep from sediments. Chemosynthetic bacteria form mats. Methanotrophic bacteria oxidize methane. Bathymodiolus mussels farm bacteria in their gills. Vesicomide clams dig into sediment, extend chemosymbiotic siphons into the seep fluids. Alvanellid worms colonize the fringes. Seeps can last thousands of years. And the mining threat is real. The Clarion-Clipperton Zone holds vast deposits of polymetallic nodules: manganese, nickel, copper, cobalt, metals for batteries, solar panels, electronics. The International Seabed Authority, under UN Convention on the Law of the Sea, issued 17 exploration contracts in the CCZ to companies and governments. Extraction would involve dragging collectors across the seafloor, sucking up nodules, discarding sediment. The impact is unknown but likely severe. The nodules take millions of years to form. Their habitat, glass sponges, bryozoans, octocorals attached to them. Removing them removes the structure the community depends on. The sediment plume from mining machines would smother animals over vast areas. A University of Hawaii study published in Limnology and Oceanography, October 2, 2025, examined the abyssal benthic boundary layer, the zone a few meters above the seafloor. Using seawater pumps on a lander, the team collected zooplankton 10 feet above the seabed in spring and fall. They found the community changes dramatically between seasons, responding to varying levels of food sinking from the surface. The animals, snails, bivalves, barnacles are sensitive to surface productivity. Deep-sea mining would disrupt that. Sediment plumes would persist for months, clogging filter feeders, reducing visibility, smothering larvae. The impact is unavoidable, regardless of time of year. The research continues. Schmidt Ocean Institute operates Falkor, the only year-round seagoing philanthropic research vessel. In April 2020, Falkor explored the Ningaloo Canyons off Western Australia. Chief scientist Nerida Wilson and colleagues from Western Australian Museum, Curtin University, Geoscience Australia, and Scripps Institution of Oceanography found a giant siphonophore, Apolemia, estimated at 45 meters long, the longest animal ever recorded. Up to 30 new species. First giant hydroids in Australia. Large communities of glass sponges in Cape Range Canyon. First observations of Taining's octopus squid, long-tailed sea cucumber and other mollusks, barnacles, squat lobsters in Western Australian deep waters. The 5 Deeps expedition in 2020 reached the deepest point in each ocean. Victor Vescovo piloted the submersible limiting factor to Challenger Deep, Mariana Trench, 10,925 meters, the deepest point on Earth. He found plastic bags and candy wrappers at the bottom. He also found life, amphipods, sea cucumbers, snailfish. Even at full ocean depth, life persists. In the Atacama Trench, east of New Zealand, the Hadal snailfish, Pseudoliperus sweari, holds the depth record for fish, 7,700 meters. It has no scales, gelatinous body, large head, small eyes. It hunts amphipods and small invertebrates. It reproduces slowly, large eggs, few offspring. It survives pressure that would liquefy shallow water fish. The adaptations pile up. Piezolites, organic molecules, trimethylamine N oxide, TMAO, that stabilize proteins under pressure. Shallow water fish have low TMAO. Deep sea fish are loaded with it. Lipid membranes with unsaturated fatty acids to maintain fluidity at cold temperatures. Enzymes with modified amino acid sequences that function at high pressure. Reduced swim bladders or no swim bladder, pressure would crush gas-filled spaces. Some fish use lipids for buoyancy instead, wax esters in liver tissue, less dense than water. Bioluminescence is ubiquitous. 90% of midwater and deep-sea animals produce light. The functions vary. Counter-illumination camouflage, lures like anglerfish, burglar alarms like vampire squid, communication, flashing patterns to signal mates, aposematism, warning coloration. Some lanternfish have species-specific photophore patterns on their bodies, mating recognition. Others use bioluminescent clouds as decoys, like squid ejecting glowing ink. The deep scattering layer, DSL, a zone in the midwater, typically 400 to 1,000 meters, packed with animals, lanternfish, Myctophidae family, hatchetfish, Sternoptychidae. They migrate daily. At night, they rise to surface waters to feed on zooplankton. At dawn, they descend to depth to avoid visual predators. This is the largest animal migration on Earth by biomass, billions of tons moving up and down every day. It was discovered during World War II when sonar operators found a layer that scattered sound, mimicking a false seafloor. The layer rose and fell with the sun. Eventually, they realized it was animals. The carbon pump depends on this. Animals feed at the surface, descend, defecate at depth. Carbon captured by phytoplankton through photosynthesis is transported to the deep ocean, where it's sequestered for centuries. The biological pump, without it, atmospheric carbon dioxide would be much higher. The abyss is a carbon sink, and temperature matters. Deep water is cold, 1.5 degrees Celsius, but it's stable. No seasons, no temperature swings. Animals don't waste energy thermoregulating. Ectotherms, body temperature matches environment. Metabolic rate is directly tied to temperature. Q10 values, the factor by which metabolic rate increases with a 10-degree temperature rise, are around 2 to 3 for most animals. For deep-sea species, the cold means slow metabolism, slow growth, slow reproduction, long lifespans. A Greenland shark can live over 400 years. The longest-lived vertebrate. Orange, roughy fish live 150 years. They don't reach sexual maturity until 20 to 30 years old. If you overfish them, recovery takes decades. The deep-sea food web is fragile. Remove one component, the system collapses. The abyss has low redundancy. Few species fill each niche. If you lose the sea cucumbers, who recycles the sediment? If you lose the sponges, where do the small animals live? The abyssal plain has been stable for tens of millions of years. Changes happen on geological timescales. Introduce rapid human disturbance, mining, trawling, climate change, and the community can't adapt fast enough. Ocean acidification reaches the abyss. Atmospheric carbon dioxide dissolves in seawater, forming carbonic acid, lowering pH. The saturation horizon, the depth at which calcium carbonate dissolves, is shoaling. It used to be below 5,000 meters. Now it's rising. Organisms with calcareous shells, foraminifera, some mollusks, corals face dissolution. The abyss is not isolated from surface changes. Oxygen minimum zones are expanding. Warmer water holds less dissolved oxygen. Deoxygenation. Vampire squids live in OMZs, but they're specialized. Most animals can't tolerate low oxygen. As OMZs expand horizontally and vertically, habitable space shrinks. Species get compressed into smaller refugia. Interactions intensify, competition increases, predation pressure rises, and microplastics infiltrate the deepest ocean. Monterey Bay Aquarium Research Institute found a fangtooth, Nuplagaster cornuta, with microplastics in its stomach. The fish had eaten a cockeyed squid and a bearded sea devil, both of which had ingested plastic. It's moving up the food chain. Plastic fibers, fragments, microbeads. They persist for centuries. Animals ingest them, thinking they're food. The plastic blocks digestive tracts. Leaches toxins, provides surfaces for pathogen colonization. The exploration technology advances, remotely operated vehicles, ROVs, like Mberi's Doc Ricketts and the NOAA Ocean Exploration ROVs. They reach 6,000 meters. Autonomous underwater vehicles, AUVs, pre-programmed missions, mapping, sampling, human-occupied vehicles, HOVs, Alvin, Shinkai, limiting factor. They carry scientists to the seafloor for direct observation. Deep-sea cameras, baited remote underwater video systems, bruvets, drop them down, wait, retrieve, landers. Stationary platforms with instruments. They sit on the seafloor for days to months, recording temperature, salinity, currents, biology. Each expedition brings new discoveries. November 2, 2025, Senckenberg Ocean Species Alliance published 14 new species descriptions. Spinther bonorum, a polycate worm. Myonera aleutiana, a carnivorous bivalve from the Aleutian Trench at 6,465 meters. It's one of the first bivalves confirmed to eat other animals. Captured prey includes nematodes, foraminifera, Valeropolina gretcheni, a mollusk from the Aleutian Trench, class Monoplacophora. Living fossils, thought extinct until 1952. Now they have a high-quality genome published from the holotype specimen. An isopod from the family Monopsidae, described as popcorn-like and parasitic. The specimens came from depths exceeding 6,000 meters, collected by submersibles and ROVs using advanced lab techniques. Light and electron microscopy, confocal imaging, molecular barcoding, micro-CT scanning. The Ocean Species Discoveries Initiative aims to speed up taxonomy. Normally, there's a decades-long delay between discovering a species and formally describing it. Many species face extinction before we even know they exist. The platform provides standardized, concise species descriptions. Senckenberg's Discovery Laboratory provides the tools. The Clarion-Clipperton Zone remains the hotspot. Over 1,500 species recorded so far. 70 to 90% are new to science. Endemism is high. Species found nowhere else. Genetic studies show limited connectivity between populations, separated by hundreds of kilometers. The abyssal plain looks homogeneous, flat mud stretching forever, but biogeographic barriers exist. Seamounts, ridges, fracture zones create isolation. Each region has unique communities. Seamounts themselves are oases, underwater mountains rising from the abyssal plain. Approximately 200,000 seamounts exist, mostly in the Pacific. They disrupt currents, create upwelling, concentrate nutrients, filter feeders, corals, sponges, crinoids, cover the slopes. Fish aggregate. Seamounts are biodiversity hotspots, but they're also targets for fishing. Bottom trawling destroys centuries-old coral gardens in minutes. Bycatch includes rare, slow-growing species. The abyss is not a frontier for exploitation. It's an ecosystem. It provides services. Nutrient cycling, carbon sequestration, genetic resources. Biomolecules from deep-sea organisms have applications in medicine, biotechnology. Heat-stable enzymes from vent bacteria are used in PCR, the foundation of genetic research. Antifreeze proteins from polar fish. Bioactive compounds from sponges with anti-cancer properties. The deep sea is a library of evolutionary solutions to extreme problems. We haven't even opened most of the books, and we're running out of time. The mining companies are waiting for permits. Contracts are issued. The technology is ready. The market demand for battery metals is surging. Electric vehicles, renewable energy storage. The pressure is economic, but the cost is biological. Once you scrape the nodules, you can't put them back. The community is gone. Extinction is permanent. Scientists argue for precautionary principle. We don't know enough. Baseline studies are incomplete. We can't predict the full impact. Conduct more research. Establish marine protected areas. Develop extraction methods that minimize harm, if extraction must happen. But the clock is ticking. Regulations are being written. Decisions are being made. March 20, 2024, the James Cook expedition documented the CCZ community, over 30 potential new species. Thomas Dahlgren from University of Gothenburg said only 1 out of 10 animal species living down there has been described by science. We're destroying habitat before we know what lives there. The abyssal plains make up more than half of Earth's surface. They're the largest ecosystem on the planet, and the least protected. No international law adequately safeguards them. The high seas, areas beyond national jurisdiction, fall into a governance gap. The International Seabed Authority regulates mining, but doesn't have conservation as a primary mandate. The UN High Seas Treaty, adopted in 2023 aims to create marine protected areas in international waters, but it hasn't been ratified. Implementation is slow. The deep sea remains vulnerable, as the irony is thick. We explore space, Mars, the moon, searching for life, but the deepest ocean, right here on Earth, is less mapped than the surface of Mars. Less than 20% of the ocean floor has been mapped to high resolution. We know more about the topography of Venus than the abyssal Pacific. The James Webb Space Telescope peers back to the dawn of the universe, but we can't see 4,000 meters down into our own ocean without expensive robots, and the creatures keep surprising us. The bigfin squid, Magnapinna, arms and tentacles reaching 8 meters long, held at right angles to the body. Elbowed, they hang vertically in the water column, dragging tentacles across the seafloor like fishing lines. Only a handful of sightings, no specimen captured intact. We don't know how they reproduce, what they eat, how long they live, just ghostly videos from ROV cameras. The colossal squid, Mesonychoteuthis hamiltoni, larger than the giant squid, hooks on its tentacles, eyes the size of dinner plates, the largest in the animal kingdom. It lives in the southern ocean around Antarctica, depths of 1,000 to 2,000 meters. In 2025, an international team aboard Schmidt Ocean Institute's research vessel, Falkor, captured the first confirmed footage of a baby colossal squid in its natural habitat. April 16, 2025. The juvenile was swimming in midwater off the coast of the Ross Sea. Transparent body, massive eyes, already a meter long. Adults reach 14 meters and weigh 750 kilograms. They hunt Patagonian toothfish and other large prey. Sperm whales dive 2,000 meters to hunt them, surface with sucker scars the size of dinner plates on their skin. The deep sea is not empty, it's not barren. It's a world as complex and diverse as tropical rainforests or coral reefs, just harder to see, harder to study, harder to love. We don't have charismatic megafauna, no polar bears, no elephants, but we have gummy squirrels and Barbie pigs and vampire squids and Dumbo octopuses. We have anglerfish with bioluminescent lures and parasitic males. We have barrel eyes with transparent heads and rotating eyes. We have giant isopods that can survive 5 years without food. We have single-celled organisms 20 centimeters across. We have siphonophores longer than blue whales. The abyss is the final frontier on Earth, and we're on the verge of irreversibly altering it. The polymetallic nodules in the Clarion-Clipperton zone took tens of millions of years to form. The communities living on them evolved in isolation, adapted to extreme stability. Remove the nodules, you remove the habitat. The sediment plume from mining machines will smother animals over thousands of square kilometers. The noise will disrupt communication, navigation, hunting. The light from work vessels will penetrate depths that have been dark for hundreds of millions of years. In June 2023, a study in Current Biology estimated 6,000 to 8,000 species await discovery in the CCZ. If mining proceeds before we've cataloged them, species will go extinct before we name them. We'll lose genetic diversity we don't understand. Evolutionary lineages that survived mass extinctions, tectonic shifts, climate changes, wiped out in a decade for metals we could recycle more efficiently from existing products. The choice is ours. The technology exists. The science is clear. We can protect the abyss or exploit it. We can study it or destroy it. The creatures down there can't speak for themselves. The gummy squirrels bouncing across the mud at 5,000 meters. The Dumbo octopuses hovering in the current at 7,000 meters. The vampire squids drifting in oxygen minimum zones. The giant isopods waiting in the cold dark for a whale carcass. They're doing what they've done for millions of years. Surviving, adapting, thriving in the most extreme environment on the planet. A soil core from the abyssal plain, 5 meters deep, every layer 1,000 years. At the bottom, microfossils from the Miocene, 20 million years old. The sediment remembers, the sediment records. If we scrape the seafloor clean, what will future sediment record? An extinction layer, a boundary marking when humanity decided the abyss was worth less than batteries. That's the story. That's what lives down there. Not monsters, not aliens, just animals solving the same problems all life faces. How to find food, avoid predators, reproduce in the darkest, coldest, most pressurized habitat on Earth. They've had millions of years to figure it out. We've had a few decades to notice, and a few years to decide their fate. Cold, dark, pressure. The abyss keeps its secrets until we look, and when we look, we find wonders.